everything I'm going to say in this particular video, you will definitely see it in your exam. Like I will always tell you, this is the right place to be. You don't need to worry, you don't need to start reading different textbooks just for you to pass biology practical. With what I'm going to say here, if you can be able to jot it down, cram it, enter the hall, believe me that you will have excellent result by the time WAEC releases this result. So if today is the first time of visiting my channel, all I want you to do is to hit the subscription button and also turn the subscription bell on, so that anytime we post in this channel, you can be able to get notification to watch our videos. So I'll be giving you breakdown on all the things you are going to see as far as this year 2025 biology practical is concerned. So all I want you to do is to get your pen and paper ready, and remember, do not skip this video, because when you skip this video, you miss one step or the other, and by the time you try to carry out your own experiment, you will have a failed result. So now let us continue. So from this specimen 2025, on the first specimen we have here, we have specimen A, which is being weevil. So now, what are the questions, what are the things that you are expected to know as far as this specimen is concerned? Here we have the kingdom. Take note, the kingdom is Animalia kingdom. Kingdom is Animalia kingdom. We have the phylum. The phylum is arthropods or the arthropoda. We have the class. The class is Incenta. Now you'll be asked the reason why it is referred to as Incenta. The reason why it is called Incenta, or it is grouped under Incentas, is because it is the only organism in the phylum of arthropods that have wings and can fly. So they have wings and can fly. That is the reason why they are referred to as Incentas. Okay. Now, the next thing there is the structure, the diagram of this particular specimen A. Go and learn how to draw it. It is already on your screen. Learn how to draw it and label it effectively. If you want to know how to draw effectively, very simple and easy, all you need to do is you watch my other video on biology practical. I gave you tutorial on the things that you are expected to do as far as drawing is concerned. Remember, drawing in biology is quite different from drawing in other part of creative and painting and all the rest of them. It's different. There are things you are expected to know. There are things you are expected to do while drawing. So go and visit that video and watch it. So that is by the way. Now on the structure of this bean beetle, it is, we have the antenna, we have the head, we have the eyes, we have the thorax, we have the leg, we have the forewings, we have the abdomen, we have the mouth part, and we have the snout, which is the mouth, the pointed mouth. Now another thing you should note here now is the feeding habit. The feeding habit. What is the feeding habit of this particular organism? Now is by the way. Now, what is their metamorphosis? What is their metamorphosis? Their metamorphosis now is complete metamorphosis, which is they undergo complete metamorphosis. They undergo complete metamorphosis, which involve the egg, the larva, the pupa, and the imago, which is the 4 stages of their life cycle. So that is why they are called the complete metamorphosis. Now, the next thing you will be asked is the economic importance. Please take note of this. Take note of, don't forget. I know why I'm saying this, because you will still come back to this channel to say that everything I said here actually came out in the exam hall. Okay, so now you'll be asked the economic importance. And remember, like I will always tell students that comes to my channel, as far as biology is concerned, whenever you hear the word economic importance means both the negative and their positive impact. So now let's take a look at the economic importance of specimen A. Now under the economic importance, here says it causes loss to stored grain. It causes loss to stored grain. 2 is it damages the viability of the beans. It damages the viability of beans. Okay. Now another one is here says it makes seed unattractive. It makes the seed unattractive. Now, the next one there says it is a vector of diseases. It is a vector of diseases. Another one there says it reduces the yield of the crop. Another one there says it increases cost of production in trying to control them. Now, the next one there says it reduces the marketable value of the crop. So due to their attack, it makes rather the crop or the seeds unattractive. And by the time it is taken to the market, you see that the value of the crop or the seed will be reduced. So those are the economic importance of these insects. Let me repeat this again. It causes loss to stored grain. It damages the viability of the beans. It makes seed unattractive. It is a vector of disease. It reduces the yield of the crop. It reduces the nutritional value of the beans. It increases cost of production in trying to control them. Now, lastly, it reduces the market value of the crop. So that is, by the way. Now, another thing there is the prevention. How do we prevent it? How do we prevent it? Now, look at this now. Before we talk about the prevention, let us go over to specimen B. Because the specimen B now is bean seed with holes in it. Now, in this specimen B, which is bean seed with holes in it, so they are just trying to look at the effect of specimen A on that of specimen B. Okay, so even in this specimen B, they may ask you what is the effect of specimen A on specimen B. Take note. What is the effect of specimen A on specimen B? So the effect of the specimen A on specimen B is now the economic importance, which is what we have just read out, which involve it causes loss to the store grains, it damages the viability of the grains, it makes it unattractive, it is a vector of diseases, it reduces the yield of the crop, it reduces the nutritional value of the beans, it increases the cost of production in trying to control the insect. Now, lastly, it reduces the market value of the crop. Take note. So that is the area where you should pay attention on. Remember, these things we are doing is a tactical work, and these are the areas where your questions will be coming from. If you are a teacher, take note. If you are a student, also take note. Okay, so now let's look at the preventive measures for specimen A and specimen B. How do we prevent the attack of specimen A, which is a bean weevil? How do we prevent the effects on specimen B? Now, we will generalize it. They are still the same thing. So in whichever one question comes in, it is the answer. Now, let's take a look at it. The use of insecticides. The use of insecticide, that is number 1. 2, proper drying before storage. When you store the crop properly when you dry the crop properly before storing it, it will prevent the attack of this particular bean weevil and also prevent the seeds from having these holes that can cause its viability. Okay. Now, another one there says destroy infected crops. So those crops that have been infected by this specimen A, you must destroy it. Possibly you burn them to avoid infecting other crops. Now, another one there says hand picking of larvae. You handpick the larvae of this particular specimen. Now, another one there says store in can bag in a cool, dry place. So all these that we have mentioned now are the preventive measures of specimen A and specimen B. Okay, I hope you get what I'm saying now. The preventive measures, we are trying to prevent this particular bean weevil, and we are trying to prevent the bean seed from having hole. Remember, what causes the hole in the bean seed is the attack of the bean weevil, which is a boring insect. Now, the bean weevil is what makes hole in the bean seed. Take note of that. So now let's move over to specimen C, which is water leaf plants. So we were asked to get the whole of water leaf plant, as the teacher will make it available on that very day, fresh one. But look at this. In this particular specimen, they will ask you to take a leaf, just a leaf, from that whole specimen. That is what you will be asked to work on. They are not going to ask you questions based on the whole part of the water leaf. No, nobody's going to do that. They will just ask you to take a leaf of it, and questions will begin to arise from that. Now, let's just look at the kingdom. Which kingdom does specimen C belong to? That is the question. Now, the specimen C is a plantae. It belongs to the kingdom of plantae. Their phylum is Trichophytes. Their botanical name is Thallium triangulae. Thallium triangulae. Okay. Now you go and learn how to draw the water leaf. Just a leaf of it. Learn how to draw it and label fully. Learn how to draw it and label fully. Okay. So now another one there says you'll be asked of the leaf type. What type of leaf does that specimen C have? The leaf is obverse. It has an obverse shape. It has an obverse shape. Okay, so we call it obverse. Now, what kind of margin does the leaf have? Does the leaf have? Okay, so the leaf has an entire, because the laminas are not broken. It doesn't have a broken lamina. So the laminas are just, together. So we say that it has an entire margin. Okay, so that is by the way. Now the next question there will be, what is the function of the leaf? What is the function of that specimen C, which you have already plucked out? Now the function is photosynthesis. Remember, leaf is responsible for photosynthesis and also for transpiration. And what do we mean by transpiration? We say that transpiration is a process by which plants loses water through their open stomata. So that is by the way. Now, another one there is they will be asking you about the venation. About the venation. And what is this venation? The venation is the arrangement of vein in a leaf. That is what we call venation. So what kind of venation does water leaf have? Now, it has a parallel venation. It has a parallel venation. It is not netted. It is not netted. It's just parallel, like this. Parallel venation. So that is by the way. Okay, so that is all for specimen C. Now let's go over to specimen D. Specimen D. The specimen D here now says quill feather. It is a quill feather. Okay, so under this specimen D, we say quill feather of a domestic fowl. Now the question you'll be asked here now is, what are the source? Where can we get this quill feather? Okay, so we can get quill feather from a duck, from chicken, from pigeon, from dove, from peacock, from eagle, and from vulture. These are the possible animals or possible possible birds where this quill feather can be gotten from. Okay. Now, what is the phylum to which this particular specimen is gotten from? The phylum to which this particular specimen is taken from or gotten from. Now, the phylum is Chordata. The kingdom is Animalae. Now, what is their class? The class is Aves. 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 Okay, so you just have to take note on that. Now, what is the adaptative features? What are the adaptative features? What is this particular feather adapted for? What is this particular quill feather adapted for? Now, the quill feather is adapted for flights, for warmth, and for courtship display. So they use the quill feather either for flights, flying, or they use it to cover their body, that is for warmth, for temperature regulation, or they use it for courtship display. By the time they open that their feather, the fellow other animals like other birds, fellow birds can be attracted to them due to the different colors that it possesses. So that is that. That's why we say courtship display, warmth, and flight. So now the next thing there is try as much as possible to learn how to draw this quill feather very, very well and label it. So on the label we have the vein, we have the rachis or the shaft, we have the barb, we have the aftershave, we have the quill. So these are the things you should take notes on. Remember, I said if you want to learn how to draw and label effectively, you can visit my other video on Biology Practical 2025. The tutorial is there. Now let's take a look at the next one, which is specimen E, known as pineapple, longitudinal section of pineapple. Now, when we talk about longitudinal section, it means that this fruit, this pineapple, will be divided into two, just the way it is on the screen. It is not going to be this way, but it is going to be divided into two, like this. And you will be asked to draw and label it fully. You'll be asked to draw and label it fully. Possibly, you'll be given scale to draw. And remember, in scale drawing, you need to make use of your meter rule. In drawing, that is why you need to visit the other video. The tutorial is there. Okay, so on this, look at the major areas you must take note on, because in your WAEC marking, there are things that are expected of, even if you do not label everything, but there are important labelings. There are important terms you must put there, which will earn you your mark. Here we have the crown cortex, we have the base of the crown, we have the ovary, we have the seed, we have the shell, we have the core, we have the fleshy, and we have the base. Very, very important. So I believe you are getting everything I am saying in this video. And remember, do not skip this video. Okay. Now, another thing that they must ask you is the placentation. What kind of placenta does this particular pineapple have? Now, the answer there is axial placentation. It has an axial placentation, which is A-X-I-L-E, axial placentation. Now, you'll be asked the fruit type. What type of fruit is specimen E? Now, the fruit type is called Sorosis. It is a Sorosis fruit, which is a fleshy fruit. It is a fleshy fruit known as Sorosis. Very, very important. Now, the mode of dispersal. What is the mode of dispersal of this particular fruit? The mode of the dispersal is man. Man is the only thing that can disperse this particular fruit. So take note of that. Now let's go over to specimen D. Now the specimen D we have here is orange fruit. The transverse section of an orange fruit. Now in this transverse section, it is no longer this way. Remember that of pineapple, which is specimen E, is longitudinal, which is this way. But this one now, the orange will be cut this way, just the way it is on the screen. Okay. So now let's take a look at some possible questions that you will be expecting. Questions that you will be expecting in the exam hall on that very day. Okay. 1. You'll be asked to draw the orange fruits. Have it in mind that the drawing will come in a transverse section, not longitudinal section, just the way you are seeing on the screen. Okay. Now, learn how to draw and label it very, very well. The drawing has the epicap, the endocap, the mesocap, the seed, the placenta, and the juice. Okay? So that is, by the way, there is no much thing there. Now, you'll be asked to state the type of fruit. What type of fruit is it? It is a berry. Okay? It is a berry fruit. Remember, it is a fleshy fruit, but also known as berry. Okay? So, why is it a berry? Because it is made up of multiple seeds. It is sweet with multiple seeds in it. Now, mode of dispersal. The mode of dispersal is man or animals. If you don't want to use man, you can use animals. Okay. Now, what is their placentation? Type of placentation. Now, they have axial placentation. They have axial placentation. Take note. Their botanical name, Citrus sinensis. That is their botanical name. Now, let's take a look at the difference. The difference between specimen E and specimen F. What is the difference between specimen E and specimen F? Now here, under specimen E, which is pineapple, we say that it is a Sorosis fruit, while specimen E is a berry. Under one there says it has scars, which is specimen E. It has scars Why this one now? There is absence of scars. Here is absence of hard seed, which is the pineapple. It has seed, but the seeds are very soft. While that of orange has a hard seed. Now, there is presence of multiple placenta. This one now, the pineapple has multiple placenta in it, while the orange fruit has just one or a single placenta. Now, the pineapple, which is specimen E, is bigger in size and structure, while this one now is smaller in size and structure, which is specimen F. Now, there is a presence of crown in specimen E. Presence of crown in specimen E, while there is absence of crown in specimen F. Yes, there is presence of core in specimen E. There is an absence of core in specimen F. So take note, these are the difference between orange and pineapple. Now let's go over to the next one, which is the similarities. Which is the similarities between E and F. What is their similarities? We say that both are fruits. Yes, we all know that both are fruits. Both have seeds. Yes. Both have either placentation or placenta. Good. Both are edible. Both are fleshy fruits. Both have sweet juice. So those are their similarities. And remember, they may not use the word similarities and difference. They may use the word compare and contrast. Now, the next one there is specimen G, which is fresh egg. Fresh egg. Now, in this particular specimen, I will be talking about both specimen G and specimen H together. So, and in this specimen G and specimen H comes with practicals. It comes with reactions, reacting chemicals with it. Because on the specimen alone, which was provided, the schools were mandated or instructed to provide some materials which are not listed among the numbers of specimen here. It only came on the instruction given to them, material that must be made available, just like every other year. But in this case, they were asked to provide sodium hydroxide, water, copper, and which other one? And egg, which is what we have here now, egg. So in this particular one, we'll be carrying out the practical. But before that, let us go over to specimen J, because we'll still come back to this. And I'll have to carry out the experiment effectively so that you will understand and see the results. Okay, now the specimen J is the longitudinal section of a boiled egg. Longitudinal section of a boiled egg with the shells intact. Okay, so what you'll be expected to do here is to learn how to draw it. You will learn how to draw an egg just the way it is on the screen. Now, the next question there is source. Which of the animals can we get this particular egg from, or this particular specimen from? Here we have duck, we have eagle, we have peacock, we have duck, and we have chicken. These are animals where this particular specimen J can be gotten from. So now let's move over to the next practical aspect of